Chapter 7, the nervous system. Here are just a handful of the learning objectives for this chapter. As you'll learn, the nervous system is pretty all-encompassing of pretty much every function of the human body. So there's pages of learning objectives, but here are the first few. List the general functions, explain the structural and functional classifications, define central nervous system and peripheral nervous system, describe the structures and functions of neurons and neuroglia, describe the general structure of the neuron and name its important anatomical regions, describe the composition of gray matter and white matter, plus many more. So generally, the nervous system is the primary control system of the body. It provides for higher mental function, emotional expression, maintenance of homeostasis, so body balance, and regulates the activities of muscles and glands. It does this via communication by the nervous system involving a combination of electrical and chemical signals. And why all body systems are influenced by the nervous system in some way. If it is to stop functioning or functions ineffectively, the body can stay alive only with the assistance of life-supporting machines. Structurally speaking, we have the central nervous system and the peripheral nervous system. The central nervous system consists of the brain and the spinal cord. They occupy the dorsal body cavity, so you know where your brain and your spinal cord are, and act as integrating and command centers of the nervous system. They interpret incoming sensory information and issue instructions based on past experience and current conditions. We'll from now on refer to it as the CNS. The peripheral nervous system, the PNS, includes all parts of the nervous system outside of the central nervous system. It consists mainly of nerves that extend from the spinal cord and brain. These spinal nerves carry the impulses to and from the spinal cord. Cranial nerves carry impulses to and from the brain. These nerves serve as communication lines, and they link all parts of the body by carrying impulses from the sensory receptors of the CNS and from the CNS to the appropriate glands and muscles. So what we're looking at here on the right is just a figure of giving us the general organization divided by structural and functional classification of the nervous system. So here at the top you have the central nervous system, directly below it peripheral nervous system. Efferent and afferent pathways we'll learn about here shortly. Sense organs for the sensory, and then our somatic and autonomic, voluntary versus involuntary. Our involuntary nervous system, the autonomic nervous system, will then split off further into the sympathetic and the parasympathetic nervous system. So we'll learn more about that later on in the lecture. Now let's expand on that functional classification, sensory division versus motor division. So the sensory division, or afferent, literally means to go toward division, consists of nerves composed of many individual nerve fibers that convey impulses to the central nervous system. So it goes towards the central nervous system. And there are sensory receptors located in various parts of the body. It just keeps the CNS informed, and somatic sensory fibers versus visceral sensory fibers. So somatic means body. So these will be found in your skin, your skeletal muscles, your joints, versus your visceral sensory fibers, which are found in your visceral organs. And then the motor division, the efferent division, exits the CNS versus afferent, which is going towards, efferent, which is exiting. So this carries impulses away from the central nervous system to affect your organs, the muscles, and the glands, to affect a motor response. We have the somatic nervous system or nervous division, which allows us to voluntarily control our skeletal muscles, often referred to as the voluntary nervous system. However, not all skeletal muscles are actively controlled by the motor division voluntarily, because there's reflexes. We have skeletal muscle reflexes rather, which are involuntary. And then the autonomic nervous system, the ANS, it regulates events that are involuntary, so we have no conscious control over them. activity of the smooth muscle, cardiac muscle, we just learned about this, they're involuntary, and the glands. This subdivision, often called the involuntary nervous system, has two parts, the sympathetic and parasympathetic, which again we'll talk about later on in the lecture, which bring about opposing effects. One stimulates, one inhibits. So let's talk about some of the supporting cells of the central nervous system. They're often lumped together, and collectively they're called neuroglia, so nerve glue, also called glial cells or glia. Neuroglia include many types of cells that support, insulate, and protect delicate neurons. And each of the different types have special functions. So the CNS neuroglia include the following: astrocytes, which they're star-shaped. They make up nearly half of all neural tissue, and that's in the top left here. They have numerous projections that have swollen ends that cling to neurons, bracing them and anchoring them to their nutrient supply lines and the blood capillaries. They form a living barrier between capillaries and neurons and help determine capillary permeability and play a role in making exchanges between the two. They help protect the neurons from harmful substances that might be in the blood. They can also help control the chemical environment in the brain by quote-unquote mopping up leaked potassium ions, which are involved in generating a nerve impulse. They can also recapture chemicals released for communication purposes. Directly below that, we have microglia. So these are spider-like phagocytes, so that monitor health of nearby neurons and dispose of debris. So functioning like a white blood cell, such as dead brain cells and bacteria. So it just goes ahead to clean those up. So it's the janitor of the glia. Below that, we have the ependymal cells. Neuroglia that line the central cavities of the brain and the spinal cord participate in the production of CSF, cerebrospinal fluid, and the beating of their cilia helps to circulate the CSF that fills those cavities and form a protective, nice watery cushion around the CNS, so around the brain and around the spinal cord. The oligodendrocytes are neuroglia that wrap their flat extensions or processes tightly around nerve fibers, the CNS nerve fibers, producing a fatty insulating covering called myelin sheaths. And lastly, here at the bottom, you can see satellite cells or Schwann cells, rather. So the supporting cells in the PNS come in these two major varieties. So the Schwann cells form the myelin sheaths around the nerve fibers of the PNS. So the oligodendrocytes form the myelin sheath of the CNS, and these form the myelin sheath of the PNS. And the satellite cells act as protective cushioning cells for peripheral neuron cell bodies. Now let's talk a little bit about neurons, otherwise known as nerve cells. They're highly specialized to transmit messages, aka nerve impulses, from one part of the body to another. Differ structurally, but have common features, so we'll go over those features in the next few slides. They have a cell body containing a nucleus and one or more slender processes extending from the cell body. So to the right, you can just see the structure of a typical motor neuron. You can see the Schwann cells that are providing that myelin sheath, the node of Ranvier, which is what separates them. So this is obviously in the PNS, right? Because the Schwann cells make the myelin sheath on the PNS. The body of that cell, the cell body, has the nucleus and the nucleolus right in the center, and then all of the projections coming off of it are the dendrites. So the cell body is the metabolic center. The rough ER is called the Nissl bodies. Neurofibrils are intermediate filaments that are important to maintaining cell shape and are abundant in the cell body. The cytoplasm contains the usual organelles except it lacks centrioles, which confirms the amitotic nature of most neurons, so they don't self-replicate. And then the processes are arm-like fibers. They vary in length. So we have dendrites, which convey incoming messages towards the cell body, whereas the axons generate nerve impulses and typically conduct them away from the cell body. So each neuron only has one axon, called the axon hillock. Axon terminals are one of the multiple ends of the axon that branch from the motor neuron axon and interact with the sarcolemma of different muscle cells to form neuromuscular junctions. So we just learned about these, and neurotransmitters as well. Each axon terminal is separated from the next neuron or its target by a tiny gap called the synaptic cleft. We also just learned about that. In such a functional junction where an impulse is transmitted from one neuron to another is called a synapse. So although they are close, remember they never touch other neurons or target cells. So this is not just in muscle cells, this is all over the body. And then myelin sheaths. So most long nerve fibers are covered with a whitish fatty material called myelin, which has a waxy appearance. Myelin protects and insulates the fibers, increases the speed of nerve impulse transmission. So you can kind of compare it to insulation that covers wires of an electrical cord. The layers keep that electrical activity flowing along that desired path. So axons outside the CNS are myelinated by Schwann cells, as previously noted, and many of these cells wrap themselves around the axon in a jelly roll fashion. Initially, the coil will be loose, but the Schwann cell cytoplasm is gradually squeezed from between the membrane layers, and when wrapping process is done, a tight coil is wrapped around the membrane, and then that myelin sheath is what encloses the axon. So most of Schwann cell cytoplasm ends up just beneath the outermost part of its plasma membrane. This part of the Schwann cell, external to the myelin sheath, is called the neurolemma, neuron husk. Because myelin sheath is formed by many individual Schwann cells, which we saw kind of on the last slide, and we'll see, you can go back to that last slide where it's like a oval rounded area and it goes down. It has gaps or indentations in between each one of those. It's almost like long beads on a necklace. Those are called nodes of Ranvier, and it occurs at regular intervals because they're all the same size. All right, just some vocab to get out of the way. So nuclei, class of organic molecules that include DNA and RNA. So pretty familiar with that. Ganglion, a group of nerve cell bodies located in the peripheral nervous system. Tract is a collection of nerve fibers in the CNS having the same origin, termination, and function. Nerve, a bundle of neuronal processes, axons outside of the central nervous system. White matter is white substance in the central nervous system, the myelinated nerve fibers, and gray matter is the gray area of the central nervous system. It contains unmyelinated nerve fibers and nerve cell bodies. Let's talk about the functional classification of neurons. So functionally, neurons are grouped according to the direction the nerve impulse travels relative to the central nervous system. So sensory neurons carry impulses from sensory receptors towards the central nervous system, while motor neurons carry impulses from the central nervous system. So sensory neurons, or afferent neurons, so afferent to go towards, these cell bodies are always found in a ganglion outside of the central nervous system, and sensory neurons keep us informed about what is happening both inside and outside of our body. The dendrite endings of sensory neurons are usually associated with specialized receptors that are activated by specific changes occurring nearby. So we'll cover some of those in the next chapter. Simple types of sensory receptors in the skin are cutaneous sense organs, and those in the muscles and tendons are referred to as proprioceptors. Pain receptors are actually bare nerve endings, and they're the least specialized of the cutaneous receptors. They're also the most numerous because pain will warn us if something bad is about to happen to our body. So it's important that we have those pain receptors. Proprioceptors will detect the amount of stretch or tension in skeletal muscles, tendons, and joints. This information is then transferred up to the brain so that proper adjustments can be made so that you maintain balance, you maintain posture. Propria comes from the Latin word meaning one's own, and proprioceptors constantly advise our brain of our own movements. So neurons that are carrying impulses from the CNS, as I just said, to the viscera, to muscles, to glands, are the motor neurons, the efferent neurons. The cell bodies of motor neurons are usually located in the central nervous system since they are carrying information from the central nervous system. This third category is interneurons or association neurons. They connect the motor and sensory neurons in neural pathways, and their cell bodies are typically located in the central nervous system as well. And here's just an image representation of some of these sensory receptors. So in A, we have those free nerve endings, which are your pain receptors, which are pretty much all over your body, right? Very important for making sure that we stay alive, that if we're doing something that's causing us pain, that we pause, stop doing it, or take a second thought. So the touch receptor, Meissner's corpuscle, you can see in B. You can see how all of these vary a little bit in the way they look, how they wrap around certain structures. C, we have the lamellar corpuscle, deep pressure receptor. So this would be how you know, deeper within your system, how you're feeling deep pressure. D, Golgi tendon, organ, proprioceptor, just like a muscle spindle, which is a proprioceptor, which we just learned about. Now let's talk about the structural classification of neurons. So we have unipolar, bipolar, and multipolar. So uni meaning one, it has a single process emerging from the cell body, as if the cell body were on a cul-de-sac, and off the main road is that axon. One of the small branches at the end of the peripheral processes are dendrites, and the remainder function as the axon, so the axon conducts impulses to and away from the cell body. These include sensory neurons of the peripheral nervous system. Bipolar has two processes, one axon and one dendrite. Rare in adults, found only in some special organs like the eyes and the nose, where they act in sensory processing as receptor cells. And then multipolar has several processes. All motor and association neurons are multipolar, and it's the most common neuron structural type in the body. So this image is illustrating what we just talked about, just in the reverse order. So you can see the multipolar neurons with those multiple processes. Down below, that bipolar neurons, one axon, one dendrite, and then unipolar, having just one short single process. Okay, so I'm going to talk a little bit about nerve impulses. I'm going to talk about it here. It's not going to be, it shouldn't be on your quiz anywhere. I'll make sure that it's not. So the plasma membrane of a resting or an active nerve is polarized, meaning that there are fewer positive ions sitting on the inner face of the neuron's plasma membrane than there are on its outer face. So in one, you see the polarized membrane is more permeable to potassium and sodium ions at rest, maintaining a more negative inside. So it has fewer positive ions on the inside compared to the outside as potassium ions exit the cell. This maintains that inactive resting state of the neuron. Two, we get a stimulus to initiate local depolarization. That stimulus will change the permeability of a local patch of the membrane, and sodium ions diffuse rapidly into the cell. This changes the polarity of the membrane. The inside becomes more positive and the outside becomes more negative at that site. Depolarization and generation of an action potential. So it's not the first time we were hearing these words. If the stimulus is strong enough, depolarization causes membrane polarity to be completely reversed. So it goes from being negative on the inside to more positive, and an action potential is initiated. So If the stimulus isn't strong enough, the cell will return to that resting membrane state and no action potential will occur. 4, propagation of action potential. So depolarization of that first membrane patch causes permeability changes in the adjacent membrane, and the events described in step 2 are repeated. Thus, the action potential propagates. So it repeats down that patch to patches adjacent. It's like a crowd doing the wave. And then repolarization. Potassium ions diffuse out of the cell as the membrane permeability changes again, restoring the negative charge on the inside of the membrane and positive charge on the outside of the surface. Repolarization occurs in the same direction as depolarization. And then 6, initial ionic conditions are restored via our handy-dandy sodium-potassium pump. The 3 sodium ions are ejected for every 2 potassium ions carried back into the cell, and now the neuron is now ready to fire again. There is a good video in section 7.2c of your ebook if you want to check that out to learn more about it. Again, you'll never see this again on a quiz or on a test in this class. Now let's talk a little bit about the transmission of signals at synapses. So you can see that first image should look vaguely similar to what we were looking at the neuromuscular junction in the last chapter. So how does the electrical impulse traveling along one neuron get across the synapse to the next neuron or effector cell to influence an activity? And the short answer is that it doesn't. Instead, a neurotransmitter chemical crosses that synapse to transmit a signal from one neuron to the next or to the target cell. So at the neuromuscular junction, like we saw in the neuromuscular junction, the way acetylcholine was used. So here in the image, right center of the slide, you can see when the action potential reaches an axon terminal, the electrical change opens calcium channels. Calcium ions in turn cause tiny vesicles containing the neurotransmitter to fuse with the axonal membrane, releasing the neurotransmitter into that synaptic cleft, into that area between the axon terminal and whatever the receiving cell is, via exocytosis. So you can see it's being held in vesicles inside the end of that axon terminal, and via exocytosis it is migrating to the membrane of that and exiting that way. The neurotransmitter molecules diffuse across the synaptic cleft and bind to receptors on the membrane of the next neuron. If enough neurotransmitter is released, the whole series of events described above, that sodium entry, that depolarization, occurs, generating a graded potential and eventually a nerve impulse in the receiving neuron around the synapse. The electrical changes prompted by the neurotransmitter binding are very brief because the neurotransmitter is quickly removed from the synaptic cleft either by diffusing away, reuptake, or enzymatic breakdown. So in chapter 6, we learned about acetylcholine and acetylcholine esterase. That's what breaks it down. This limits the effect of each nerve impulse to a period shorter than a blink of an eye. Again, while a lot of this stuff is very interesting, I don't think any of your quiz questions are on this, and it won't be on your final exam. I don't think there's a video in your ebook about this, but do take note that the transmission of an impulse is an electrochemical event. So transmission down the length of the neuron's axon is basically electrical, but the next neuron is stimulated by a neurotransmitter, which is chemical. So it's both. It's electrochemical. Different neurotransmitters are released in different parts of the body, depending on quote unquote who's talking and what is trying to be expressed. Now let's talk about reflexes. So reflexes are rapid, predictable, and involuntary responses to stimuli. A reflex arc is a neural pathway involving both the central nervous system and the peripheral nervous system structures. It has a minimum of 5 elements: the receptor, effector, sensory and motor neurons, and the CNS integration center. So there are somatic reflexes, which stimulate the skeletal muscles. These are involuntary reflexes. even though skeletal muscles normally are under voluntary control. So this is like when you quickly pull your hand away from something hot, that's a somatic reflex. Autonomic reflex, regulate activity of smooth muscles, the heart and the glands, again, involuntary. The secretion of saliva, changes in the size of your eye pupils, so your pupillary reflex, that constriction or dilation of your pupils. Autonomic reflexes regulate such body functions as digestion, elimination, blood pressure, and sweating. So you can see why some of these things are good, that they are involuntary. On this slide, we're looking at some simple reflex arcs. So we talked about those 5 basic elements: the receptor, effector, sensory motor neuron, and the integration center. So we can see the patellar or knee-jerk reflex, which I'm sure all of you have had done in a basic physical. The patellar reflex, in which the quadriceps muscles attached to the hip tendon are stretched, is familiar. It's used for testing just general health during a physical exam. Most reflexes are much more complex than that 2-neuron reflex, involving synapses between one or more interneurons in the CNS integration center. So the flexor or withdrawal reflex is a 3-neuron reflex in which a limb is withdrawn from a painful stimulus. So a 3-neuron reflex arc also consists of 5 elements: receptor, sensory neuron, interneuron, motor neuron, and effector. Because there is always a delay in synapses, it takes time for neurotransmitter to diffuse through the synaptic cleft. The more synapses there are in a reflex pathway, the longer the reflex takes to happen. So many spinal reflexes involve only spinal cord neurons and occur without brain involvement. So these are the kind of reflexes that when we're testing somebody for possible brain death, we're testing their spinal cord reflexes versus their cranial nerves, because one will show us that their brain is working, one will show us that we're just getting reflexes from the spine. So development in regions of the human brain. So the brain can be considered in terms of 4 main parts. We have cerebral hemispheres, diencephalon, brainstem, and cerebellum. In a developing brain, as you can see on the left, the cerebral hemispheres are smooth initially, forced to grow posteriorly and laterally over the other brain regions by the bones of the skull. In the adult brain on the right, the cerebral hemispheres are now highly convoluted, enclose the diencephalon in the superior part of the brainstem. The left cerebral hemisphere is drawn so that it looks transparent, so you can see. Obviously, in a human brain, it does not look like that, but that's just so you can see the diencephalon and the superior part of the brainstem. Now let's just talk about the functions of those 4 major brain regions, starting with the cerebral hemisphere. So the cortex of the cerebral hemispheres with gray matter localizes, interprets sensory inputs, controls voluntary and skilled skeletal muscle activity, and acts in intellectual and emotional processing. In the basal nuclei of the cerebral hemispheres is the subcortical motor centers that help control skeletal muscle movements. In the diencephalon, we have the thalamus, the hypothalamus, and the limbic system, or rather, the hypothalamus is part of the limbic system. The thalamus relays sensory impulses to the cerebral cortex, relays impulses between cerebral motor cortex and lower motor centers and involved in memory, while the hypothalamus is the chief integration center of the autonomic or involuntary nervous system, regulates body temperature, food intake, water balance, and thirst. It regulates hormonal output of the anterior pituitary gland and acts as an endocrine organ producing ADH, antidiuretic hormone, and oxytocin. And the limbic system is a functional system that includes the cerebral and diencephalon structures, the hypothalamus and the anterior thalamic nuclei, mediates emotional response involved in memory processing. The midbrain of the brainstem contains visual and auditory reflex centers, contains subcortical motor centers, and contains nuclei for cranial nerves 3 and 4, contains projection fibers in the pons of the brainstem, relays information from the cerebrum to the cerebellum, cooperates with medullary centers to control respiratory rate and depth, contains nuclei of cranial nerves 5 through 7, and contains projection fibers. The medulla oblongata of the brainstem relays ascending sensory pathway impulses from skin and proprioceptors, contains nuclei that control heart rate, blood vessel diameter, respiratory rate, vomiting, etc., relays sensory information to the cerebellum, contains nuclei of cranial nerves 8 through 12, and contains projection fibers, and site of crossover of pyramids. The reticular formation is a functional system of the brainstem and maintains cerebral cortical alertness, filters out repetitive stimuli, and helps regulate skeletal and visceral muscle activity. So that filtering of the repetitive stimuli is pretty important for just being able to live. You know, we are constantly bombarded by stimuli, whether that be sound, light, touch, you know, and if our body is responding to every single stimuli, it's going to be very inefficient. And the cerebellum processes information from the cerebral motor cortex, proprioceptors, and visual and equilibrium pathways, provides instructions to cerebral motor cortex and subcortical motor centers, resulting in smooth, coordinated skeletal muscle movements. It's responsible for proper balance and posture. So just a little reiteration here. We have the cerebral hemispheres together making the cerebrum. There's 2 cerebral hemispheres. It's the most superior part of your brain, has elevated ridges called gyri, and they're separated by sulci or sulcus, which is a singular form of that. Deeper grooves, which are less numerous, are called fissures and separate large regions of the brain. The hemispheres are separated by a single deep fissure called a longitudinal fissure. Other divisions are by sulci and fissures are called lobes. Each hemisphere has 3 basic regions: a superficial cortex of gray matter, internal area of white matter, and basic nuclei. Now let's talk about the cerebral cortex. So outer gray matter of the cerebrum, speech, memory, logical and emotional responses, consciousness and interpretation of sensation and voluntary movement. Primary somatic sensory area is located in the parietal lobe posterior to the central sulcus. This is where impulses traveling from the body's sensory receptors, except for the special senses, which are in Chapter 8, are localized and interpreted. Allows us to recognize pain, differences or changes in temperature, and light touch. Primary motor area is anterior to the central sulcus, and the frontal lobe allows us to consciously move our skeletal muscles. Axons of these motor neurons form major voluntary motor tract, the pyramidal tract or corticospinal tract, which descends to the spinal cord. So what you're looking at here is basically a map. You can see from the top the cross-section cut. It's called a homunculus. We have a motor map in the precentral gyrus and the sensory map in the postcentral gyrus. The relative amount of cortical tissue devoted to each function is indicated by the amount of gyrus occupied by the body area diagram. The primary motor cortex is shown on the left side of the diagram, and the somatic sensory cortex is on the right. You can just kind of give this a look, and this is a very interesting way for them to make it so that you can kind of see which parts of the brain are related to which parts of your external body. Also contained in the cerebral cortex is Broca's area, which is the specialized cortical area that is very involved in our ability to speak, aka the motor speech area. This sends motor signals to our mouths. Anterior association area is involved in higher intellectual reasoning and socially acceptable behavior. Anterior part of the frontal lobes. Frontal lobes are also associated in language comprehension. There is some research, a lot of research, that's indicating that you don't have full frontal lobe development until early to mid 20s, which is sometimes some of the decisions we make at that age, some of the things we do and say are maybe not the most appropriate. And the posterior association area encompasses part of the posterior cortex and plays a role in recognizing patterns and faces and blending several different inputs into one understanding of a whole situation. There is a speech area here as well that allows us to sound out words. So like Broca's area, the speech area is located only in one hemisphere. Most of the remaining cerebral hemisphere tissue is the deeper cerebral white matter. It's composed of fiber tracts carrying impulses to, from, and within the cortex. So one very large fiber tract, the corpus callosum, connects the cerebral hemisphere. So these fiber tracts, such fiber tracts that, like the corpus callosum, are called commissures. The corpus callosum arches above the structures of the brainstem and allows the cerebral hemispheres to communicate with one another. And this is important because, as we already noted, things like Broca's area and other cortical function areas are only located in one hemisphere. So we have to let those hemispheres communicate. Association fiber tracts connect areas within a hemisphere, while projection fiber tracts connect the cerebrum to the lower central nervous system centers, such as the brainstem. Although most of the gray matter is in the cerebral cortex, there are several islands of gray matter called the basal nuclei that are buried deep within the white matter of the cerebral hemispheres. The basal nuclei will help regulate voluntary motor activities by modifying instructions, particularly in relation to starting or stopping movement, sent to skeletal muscles by the primary motor cortex. A tight band of projection fibers called the internal capsule passes between the thalamus and the basal nuclei. So what we're looking at here is a frontal section, so facing posteriorly, of the brain, showing the commissural association and projection fibers running through the cerebrum and the lower CNS. So you can see that corpus callosum right there in the center, right above the brainstem, that is connecting the hemispheres. You can see the basal nuclei, the longitudinal fissure down the center that separates those two, the intersection of the projection fibers, the dura oblongata at the bottom, just inferior to the pons, the thalamus, the purple right in the center, and yeah, this is a good image to show you the way those fibers interact and how they're distributed throughout the brain. Now let's talk about the diencephalon. So this is the interbrain. It sits on top of the brainstem. It is enclosed by the cerebral hemispheres. Its major structures include the thalamus, the hypothalamus, and the epithalamus. The thalamus is enclosed in the shallow third ventricle of the brain and is a relay station for sensory impulses passing upward to the sensory cortex. The hypothalamus, hypo, under the thalamus, makes up the floor of the diencephalon. Important autonomic center, as it has a role in regulating body temperature, water balance, metabolism. It is the center for many drives and emotions and an important part of the limbic system. So the emotional visceral brain. It regulates the pituitary gland and produces hormones. And the epithalamus forms the roof of the third ventricle, pineal gland, choroid plexus, which is knots of capillaries within each of the four ventricles, and along with the ependymal cells, which we learned about a few slides back, lining the ventricles. It helps to form the CSF. The pineal gland that's part of the epithalamus is also part of the endocrine system, much like the pituitary gland that is regulated by the hypothalamus. So the brainstem is about the size of the thumb in diameter and approximately 3 inches long, or 7.5 centimeters. Its structures include the midbrain, the pons, and the medulla oblongata. In addition to providing a pathway for ascending and descending tracts, the brainstem has many small gray matter areas, nuclei. These nuclei produce a rigidly programmed autonomic behaviors necessary for survival. So some are associated with the cranial nerves and control vital activities such as breathing and blood pressure. And then we'll go ahead and talk more about those different brainstem areas. So let's start with the midbrain and the pons of the brainstem. So a relatively small part of the brainstem is the midbrain, which extends from the mammillary bodies to the pons inferiorly. The cerebral aqueduct is a tiny canal that travels through the midbrain and connects the third ventricle of the diencephalon to the fourth ventricle below. Anteriorly, the midbrain is composed primarily of two bulging fiber tracts, the cerebral peduncles, literally meaning little feet of the cerebrum, which convey ascending and descending impulses. Dorsally located are four rounded protrusions called the corpora quadrigemina, because they reminded some anatomists of two pairs of twins. These bulging nuclei are reflex centers involved with vision and hearing. The pons is a rounded structure that protrudes just below the midbrain. It means bridge, and this area of the brainstem is mostly fiber tracts, aka bundles of nerve fibers in the central nervous system. However, it does not have important nuclei involved in control of breathing. So the medulla oblongata is the most inferior part of the brainstem. It merges into the spinal cord below without any obvious structure change. So like the pons and the medulla is an important fiber tract area. It is an area where important pyramidal tracts or motor fibers cross over to the opposite side, which you could see on that brain cross-section a few slides back. The medulla also contains many nuclei that regulate vital visceral activities. It contains centers that control heart rate, blood pressure, breathing, swallowing, and vomiting, among other things. And the fourth ventricle lies posterior to the pons and the medulla and anterior to the cerebellum. So reticular formation, it extends the entire length of the brainstem and is a diffuse mass of gray matter. Neurons of the reticular formation are involved in motor control of the visceral organs, for example, controlling the smooth muscle of the digestive tract. A special group of reticular formation neurons called reticular activating system, the RAS, play a role in consciousness and the awake-sleep cycle. The RAS also acts as a filter for the flood of sensory inputs that stream up the spinal cord and brainstem daily. Weak or repetitive signals are filtered out, but unusual or strong impulses do reach consciousness. Damage to this area can result in prolonged unconsciousness, aka a coma. So this image is just a mid-sagittal section of the brain, illustrating the diencephalon in the purple and the brainstem in the green. You can see the medulla oblongata, which goes right into that spinal cord, directly superior and anterior to that is the pons, and you can see the peduncles, the cerebral aqueduct, the corpora quadrigemina of the midbrain. You can see the hypothalamus, the thalamus, and the pineal gland, as well as the choroid plexus here, just posterior to the brainstem, which is responsible for, with the help of those ependymal cells, of making the cerebral spinal fluid that cushions the brain and the spinal cord. So the reticular formation extends the length of the brainstem, as I just said. The ascending arrows, so those that are pointing up, indicate sensory input to the cerebrum, and the descending arrows indicate efferent output to the reticular neurons. So you can see here we have visual impulses coming through the eye, going down to the reticular neurons, ascending general sensory tracts like touch, pain, and temperature, the descending motor projections to the spinal cord, auditory impulses, and radiations to the cerebral cortex. Then we have the large cauliflower-like cerebellum. It projects dorsally from under the occipital lobe of the cerebrum. Like the cerebrum, the cerebellum has two hemispheres and a convoluted surface, so has all those valleys, rather. Like the cerebrum, the cerebellum also has an outer cortex made up of gray matter and an inner region of white matter. It provides the precise timing for skeletal muscle activity, controls your balance. Thanks to its activity, body movements are smooth and coordinated. It plays a role less well when it's sedated by things like alcohol. So fibers reach the cerebellum from the equilibrium apparatus of the inner ear, the eye, proprioceptors of skeletal muscle and tendons, and many other areas. It can be compared to an autopilot, continuously comparing the brain's intentions with actual body performance by monitoring body position and the amount of tension in various body parts. When needed, the cerebellum sends messages to initiate the appropriate corrective measures. And in lecture, we'll talk a little bit about a way we can test cerebellar function. Now let's talk a little bit about how the central nervous system is protected. We have meninges, cerebrospinal fluid, and the blood-brain barrier. So the three connective tissue membranes covering and protecting the central nervous system structures, so that would be your brain and your spinal cord, are the meninges. The outermost layer is the leathery dura mater, meaning tough and hard mother. It is double-layered membrane where it surrounds the brain. One of its layers is attached to the inner surface of the skull, forming the periosteum or the periosteal layer. The other, called the meningeal layer, forms the outermost covering of the brain and continues as the dura mater of the spinal cord. The dural layers are fused together except in three areas where they separate to enclose the dural venous sinuses that collect venous blood, such as the superior sagittal sinus. In several places, the inner dural membrane extends inward to form a fold that attaches the brain to the cranial cavity. Two of these folds, the falx cerebri and the tentorium cerebelli, separate the two cerebral hemispheres and the cerebellum from the cerebrum, respectively. The middle layer is called the arachnoid matter. Arachnida means spider, and some think of the arachnoid membrane. They named it that because they think it looks like a cobweb. Its thread-like extensions span the subarachnoid space to attach to the innermost membrane and the pia mater, gentle mother. The delicate pia mater clings tightly to the surface of the brain and the spinal cord following every fold, so it's like a shrink wrap. The subarachnoid space is filled with cerebral spinal fluid. Remember, choroid plexuses produce the CSF, and specialized projections of the arachnoid membrane, called the arachnoid granulations, protrude through the dura mater. The cerebral spinal fluid is absorbed into the venous blood in the dural sinuses through the arachnoid granulations. Cerebrospinal fluid, CSF. So again, choroid plexuses continually form CSF from blood. It's like a watery broth with components similar to blood plasma, from which it forms, just containing less protein and more vitamin C. Inside the brain, CSF is continually moving, which is demonstrated in the image on the right. It circulates from two lateral ventricles in the cerebral hemispheres into the third ventricle in the Diencephalon, and then through the cerebral aqueduct of the midbrain into the fourth ventricle dorsal to the pons and medulla oblongata. Some of the fluid reaching the fourth ventricle simply continues down to the spinal cord, but most often it circulates into the subarachnoid space through 3 openings, the paired lateral apertures and the median aperture. So aper means open. The CSF returns to the blood in the dural venous sinuses through the arachnoid granulations. In this way, CSF is continually moving, continuously replaced. Ordinarily, so in a healthy person with no issues, CSF forms and drains at a constant rate so that a normal pressure is maintained in the skull, and a normal volume is maintained, which is about 150 ml, so about a half cup. Any significant changes in composition of CSF, blood cells, could indicate disease processes like meningitis, other brain pathologies such as tumors or multiple sclerosis, and those are determined by a procedure called a lumbar spinal puncture, in which a sample of CSF is obtained. Because withdrawal of fluid decreases CSF fluid pressure, the patient must remain horizontal for about 6 to 12 hours after the procedure to prevent what's been considered an agonizingly painful spinal headache. Lastly, let's talk about the blood-brain barrier. So no other body organ is so absolutely dependent on a constant internal environment as the brain. So other body tissues can withstand small fluctuations in the concentration of things like hormones, ions, nutrients, particularly after things like eating and exercising. But if the brain were exposed to those chemical changes, uncontrolled neural activity might result. So because certain ions like sodium and potassium are involved in initiating nerve impulses, and some amino acids serve as neurotransmitters. Consequently, neurons are kept separated from blood-borne substances by the blood-brain barrier, composed of the least permeable capillaries in the entire body. These capillaries are almost seamlessly bound together by tight junctions. So we learned about this, I think, in chapter 3. So these junctions are kind of like duct tape, tape to duct tape. Of water-soluble substances, only water, glucose, and essential amino acids pass easily through walls of these capillaries. Metabolic wastes, such as urea, toxins, proteins, and most drugs, are prevented from entering brain tissue. Non-essential amino acids and potassium ions not only are prevented from entering the brain, but also are actively pumped from the brain into the blood across capillary walls. Although the bulbous feet of the astrocytes that cling to the capillaries may contribute to the barrier, the relative impermeability of the brain capillaries is the most responsible for providing this protection. Unfortunately, the blood-brain barrier is virtually useless against fats, respiratory gases, and other fat-soluble molecules that diffuse easily through all plasma membranes. This explains why blood-borne alcohol, nicotine, and anesthetics can affect the brain. I want to briefly cover some brain dysfunction. So obviously there is a huge array of them. Your book goes over traumatic brain injuries and cerebrovascular accidents. So traumatic brain injuries are the leading cause of accidental death in the United States. So this could be from a car crash, a bike crash, just some kind of accident in general. So a concussion occurs when brain injury is slight. So person might become dizzy, quote unquote, see stars, may have a loss of consciousness briefly, but typically little permanent brain damage occurs. Generally it's just watchful waiting. We ask them to rest. If you have any vomiting, to go to the ER to get a CT, that kind of thing. A brain contusion results from marked tissue destruction. So it's like a bruise on your brain. So if the cerebral cortex is injured, the individual may remain conscious, but severe brainstem contusions may result in a coma lasting from hours to a lifetime due to injury of the reticular activating system, so the RAS. After head blows, death may result in intracranial hemorrhage, so ICH, from intracranial hemorrhage, bleeding from that ruptured vessel, or cerebral edema, swelling of the brain due to the inflammatory response of the injury. Individuals who are initially alert and lucid following head trauma and then begin to deteriorate neurologically are most likely hemorrhaging or suffering the delayed consequences of edema, both of which compress vital brain tissue and require immediate medical interventions. And then cerebral vascular accidents shortened to CVA, aka strokes, fifth leading cause of death in the United States. Usually occur when blood circulation to the brain is blocked, as by a clot or a ruptured blood vessel, and vital brain tissue dies. So this here is describing an ischemic stroke. So ischemia is occurring of the brain tissue. Brain tissue is dying. There is such a thing that it does not talk about here called a hemorrhagic stroke, which is just like it sounds. Something is, something has burst, something is hemorrhaging in the brain, causing inadequate flow of oxygen and blood throughout the brain. So we'll just talk about ischemic strokes here. So impairment will depend on severity and location. This is one of the things where you have to act pretty quickly in order to correct the issue if it's correctable, whether that be by clotbuster, TPA, or by a surgical procedure to pull the clot out. There are many. It may cause things like hemiplegia, so one-sided paralysis. But that would mean, so let's say if I have left hemiplegia, it means the right motor cortex of my frontal lobe is most likely involved, because those sensory paths cross. So your right motor cortex is responsible for your left body moving. Aphasias are also a common result of damage to the left cerebral hemisphere where the language areas are located. So we have many types of aphasias. Most common is motor aphasia, which we learned about Broca's area. And it will cause a loss of the ability to speak, and sensory aphasia, in which a person loses the ability to understand written or spoken language. Aphasias are very frustrating for the patient because, as a general rule, their intellect is unimpaired. They know what they want to say. They understand what you're saying sometimes, but they just can't form the words to respond back to you. If you've ever seen a patient with this, it is truly frustrating for them. Brain lesions can also cause marked changes in a person's disposition. In such cases, like a tumor as well as a CVA might be suspected. Some patients recover at least part of their lost faculties because undamaged neurons spread into areas where neurons have died and take over some lost functions. Most of the recovery seen after brain injury is due to this phenomenon. So not all strokes are completed, and that's called, it's just temporary brain ischemia, a transient ischemic attack. It can cause a lot of those same initial symptoms though, like numbness, temporary paralysis, impaired speech. Although these defects are not permanent, they do constitute red flags that warn of impending and more serious CVAs. This is when we start evaluating this patient for underlying causes of the TIA, because it might be leading to a bigger CVA. So we spent quite a bit of time on the brain. Let's move on to the other part of the nervous system, the spinal cord. So the cylindrical spinal cord is a glistening white continuation of the brainstem. The spinal cord provides a two-way conduction pathway to and from the brain. It is the major reflex center. Spinal reflexes are completed at this level. It's enclosed in the vertebral column that we learned about when we learned about the skeletal system, and it extends from the foramen magnum of the skull to the first and second lumbar vertebra, where it ends just below the ribs. Like the brain, the spinal cord is cushioned and protected by meninges. The meningeal coverings do not end at the second lumbar vertebra, but instead extend well beyond the end of the spinal cord in the vertebral canal. Because there is no possibility of damaging the cord below L3, the meningeal sac inferior to that point provides a nearly ideal spot for a lumbar puncture to remove CSF for testing. So in humans, there are 31 pairs of spinal nerves arising from the cord and exiting from the vertebral column to serve the body area close by. The spinal cord is about the size of a thumb for most of its length, but it is enlarged in the cervical and lumbar regions where the nerves serving the upper and lower limbs arise and leave the cord. Because the vertebral column grows faster than the spinal cord does not reach the end of the vertebral column, and the spinal nerves leaving the inferior end must travel through the vertebral canal for some distance before exiting. This collection of spinal nerves at the inferior end of the vertebral canal is called the cauda equina because it looks like a horse's tail. So this is just the anatomy of the spinal cord from the posterior view. So the spinal cord begins to descend down through the vertebral column, starting at the brainstem, and you can see where it ends. The cauda equina here, it does look like a horsetail at the bottom. We have the cervical spinal nerves, thoracic spinal nerves, and the lumbar spinal nerves, and also the end of the spinal cord. So the nerves go past the end of the cord itself. So gray matter of the spinal cord and the spinal roots. It looks like we'll see in the next slide an image of it, but it looks like a butterfly with the letter H in the cross section of the spinal cord. It has dorsal horns or posterior horns and ventral horns or anterior horns. Surrounds the central canal, which contains CSF, and neurons with specific functions are found here. So the dorsal horns contain interneurons in the dorsal root ganglia, and the ventral horns contain cell bodies of motor neurons of the somatic nervous system, which send their axons out the ventral root of the cord. Dorsal and ventral roots fuse to form the spinal nerves. So you can see there in the center, butterfly shaped is the gray matter. You can see the posterior horns are the two points on either side. There's actually also a lateral horn, and you can see the central canal right in the middle where the CSF is. We can also see the dorsal root ganglion and the dorsal root of the spinal nerve, ventral root. So the ventral and the dorsal will come together to form that spinal nerve, which you can see all the way to the left. Now let's talk about the white matter of the spinal cord. So basically everything else around that little butterfly. It's composed of myelinated fiber tracts divided into three regions: the dorsal column, the lateral column, and the ventral column. Each contains a number of fiber tracts made up of axons with the same destination and function. It has sensory afferent tracts and motor efferent tracts. All tracts in the dorsal column are ascending tracts that carry sensory input to the brain, and the lateral and ventral columns contain both ascending and descending tracts. So this image can seem pretty busy, but it's just the schematic of ascending or sensory and descending motor pathways between the brain and the spinal cord. So again, white matter of the spinal cord is composed of those myelinated fiber tracts, some running to higher centers, some traveling from the brain to the cord, and some conducting impulses from one side of the spinal cord to the other. And that's what this image is illustrating. So now let's talk about the basic structure of a nerve. So a nerve is a bundle of neuron fibers found outside of the CNS. Within a nerve, neuron fibers or processes are wrapped in protective connective tissue coverings. So again, these are acting like the wrapping of an electrical cord so that the impulse is stays contained in the direction that you want it to go. Each fiber is surrounded by a delicate connective tissue sheath called the endoneurium. Groups of fibers are bound by a coarser connective tissue wrapping called the perineurium, and to form fiber bundles or fascicles. Finally, all of the fascicles are bound together by a tough fibrous sheath called the epineurium to form the cord-like nerve, which you can see here to the right. First we have the axon, the myelin sheath, so made of the Schwann cells. Each of those is covered by endoneurium. A bundle of those is covered by perineurium, making a fascicle. And then Fascicles are bound together by the epineurium. There are 12 pairs of cranial nerves primarily serving the head and neck. Only one pair, the vagus nerves, extends to the thoracic and abdominal cavity. They're numbered in sequence. Most cases, the names will reveal the important structures they control. So we'll start with one. So the olfactory nerve. It arises from the olfactory receptors in the nasal mucosa and synapse with the olfactory bulbs. It's purely sensory, carries impulses for the sense of smell. So in order to test this, you would ask the client or the patient to sniff something to identify something like cloves or vanilla. I will say generally in a hospital setting, this is something we defer, but oftentimes I've seen people use an opened alcohol swab. Then we have the optic nerve. So these fibers arise from the retina of the eye and form the optic nerve. The two optic nerves form the optic chiasma by partial crossover of the fibers, and the fibers continue to the thalamus as the optic tracts. The lambic fibers connect to the visual cortex. So this is also purely sensory, carries impulses for vision, and you test this by testing vision via things like the Snellen chart and visual field tests by moving your finger in and out of a patient's field of vision. Number 3 is an ocular motor. So that's already telling you eye and movement. So fiber runs from the midbrain to the eye. This is purely motor. It supplies motor fibers to 4 of the 6 muscles, superior, inferior, medial rectus, and inferior oblique, that direct the eyeball to the eyelid and to the internal eye muscles controlling lens shape and pupil size. So to assess for this, pupils are examined for size, shape, and size equality. Pupillary reflex is tested with a pen light, so that's when somebody rudely shines a light in your eye to see if your pupils constrict. Eye convergence is tested as well as the ability to follow moving objects. So follow the tip of my pen with just your eyes, not your head. 4, the trochlear. This fiber runs from the midbrain to the eye, also purely motor. Supplies motor fibers for 1 external eye muscle, and that is the superior oblique eye muscle. Tested in common with cranial nerve 3 for the ability to follow moving objects. The trigeminal. So fibers emerge from the pons and form 3 divisions that run to the face. It's a mixed nerve, so it conducts sensory impulses from the skin of the face and the mucosa of the nose, mouth, and corneas, and also contains motor fibers that activate the chewing muscles. So trigeminal, it shows us sensations of pain, touch, and temperature. Things like corneal reflex is tested with a wisp of cotton, and the motor branch is tested by asking a subject to open mouth against resistance and move jaw up and down and side to side. So obviously you wouldn't inflict pain on patients, hopefully, but you could start with touch, and if they can't distinguish touch or can't feel touch, then you might have to use a more noxious stimulus. 6, the abducens. So this fiber leaves the pons and runs to the eye. This is also purely motor. Supplies motor fibers to the lateral rectus muscle, which rolls the eye laterally. Tested in common with cranial nerve 3 for the ability to move each eye laterally. 7 is the facial nerve. Fibers leave the pons and run to the face. It's a mixed nerve as well. Activates the muscles of facial expression and the lacrimal and salivary glands. It also carries sensory impulses from the taste buds of the anterior tongue. So anterior 2/3 of the tongue is tested for its ability to taste sweet, salty, sour, and bitter. Subject is asked to close eyes, smile, whistle, etc. Tearing of eyes is tested with ammonia fumes. We've never done this, I've never done that in practice. But 8, vestibulocochlear. Fibers run from the equilibrium and hearing receptors of the inner ear to the brainstem. It is also a mixed nerve. Vestibular branch transmits impulses for the sense of balance, and the cochlear branch transmits impulses for the sense of hearing. Small motor components adjust sensitivity of sensory receptors. So hearing is checked by air and bone conduction using a tuning fork. Oftentimes in practice, if you don't have a tuning fork, you can just do a whisper test where you stand approximately 3 to 5 feet to the side or behind a patient and whisper 3 words and see if they can hear you. 9, glossopharyngeal. Fibers emerge from the medulla and run to the throat. also a mixed nerve, supplies motor fibers to the throat or pharynx that promote swallowing and saliva production, carries sensory impulses from the taste buds of the posterior tongue and from chemical and pressure receptors of the carotid arteries. So gag and swallowing reflexes are checked. So if subject can speak, if your patient can speak, you just ask them to cough. Posterior tongue may be tested for taste. 10, the vagus nerve. So fibers emerge from the medulla and descend into the thorax and abdominal cavity. Also a mixed nerve. Fibers carry sensory impulses from and motor impulses to the pharynx, larynx, and abdominal and thoracic viscera. Most fibers are parasympathetic fibers and promote digestive activity and help regulate heart activity. So tested in common with cranial nerve 9 because they both serve muscles of the throat. 11, accessory. Fibers arise from superior spinal cord C1 to C5 and travel to muscles of the neck and back, purely motor, activating the sternocleidomastoid and the trap muscles. So checked by checking these muscles, asking the subject to rotate their head or shrug their shoulders against resistance. And then lastly, the hypoglossal. Fibers run from the medulla to the tongue. Purely motor fibers control tongue movements. You ask your patient to stick out their tongue and move it from side to side, and it'll help you determine if there's any abnormalities. As we learned previously, spinal nerves, there are 31 pairs of human spinal nerves, and they're formed by joining the ventral and dorsal roots of the spinal cord. Although each of the cranial nerves issuing from the brain is named specifically, the spinal nerves are named for the region. They divide into the dorsal ramus and the ventral ramus, making each one about half an inch long, and plexuses are networks of nerves formed by joining ventral rami of spinal nerves containing both sensory and motor fibers. Here are just a few examples of some spinal nerve plexuses. Not going to require you to memorize these, but just know that it's a complex network of nerves in each area. They have an origin. They have multiple important nerves generally that each serve certain body areas, and then damage to the plexus or its nerves will affect certain things. So for example, not shown here, we have the cervical plexus, which is C1 to C5, and important nerves include the phrenic nerve. It serves the diaphragm, which is very helpful and responsible for inspiration and expiration of air in and out of your lungs, and it serves the skin and muscles of the shoulder and neck. So you imagine if this plexus is damaged, it can cause respiratory paralysis and death if not promptly treated. So I encourage you to go look at table 7.3 in reference to this image as well, to kind of see what each of these plexuses control and affect. So let's talk about the autonomic nervous system, which is the motor subdivision of the peripheral nervous system. It controls body activities automatically. It is composed of a specialized group of neurons that regulate cardiac muscle, smooth muscle, and glands. Although all body systems contribute to homeostasis, the relative stability of our internal environment depends largely on the workings of the autonomic nervous system. At every moment, signals flood the visceral organs into the CNS, and the autonomic nervous system makes adjustments as necessary to best support body activities. For example, blood flow may be shunted to more needy areas. Heart and breathing rate may be sped up or slowed down. Blood pressure may be adjusted. Stomach secretions may be increased or decreased. Most of this fine-tuning occurs without our awareness or attention, and few of us realize our pupils dilate or our arteries constrict. Hence, the autonomic nervous system is also called the involuntary nervous system. So, when we're comparing the autonomic nervous system and the somatic nervous system, besides differences in their effector organs and in the neurotransmitters they release, the patterns of their motor pathways differ. So you can see in the somatic division, the cell bodies of the motor neurons are inside the central nervous system, and their axons in spinal nerves and their rami extend all the way to the skeletal muscles they serve. The autonomic nervous system, however, has a chain of 2 motor neurons. The first motor neuron of each pair, the preganglionic neuron, is the brain or spinal cord. Its axon is the preganglionic axon, and it leaves the CNS to form a synapse with the second motor neuron and the ganglion outside of the central nervous system. The axon of this ganglionic neuron, the postganglionic axon, then extends to the organ it serves. The autonomic nervous system has 2 arms, the sympathetic and the parasympathetic. Both serve the same organs, but cause opposite effects. The sympathetic division mobilizes the body during extreme situations, fear, exercise, rage, whereas the parasympathetic division allows us to unwind and conserve energy. We examine these differences in more detail shortly, but you can see it illustrated here, the different neurotransmitters at the effector organs, and how a single neuron from the CNS to the effector organs is how the somatic nervous system works, and there's a 2 neuron chain from the CNS to the effector organs in the autonomic nervous system. So here we're just looking at the anatomy of the autonomic nervous system. We have the parasympathetic and the sympathetic. Parasympathetic fibers are shown in purple and sympathetic fibers in green. Solid lines represent preganglionic fibers, and dashed lines indicate postganglionic fibers. Note the direct connection between the sympathetic nervous system and the adrenal gland. So remember, the sympathetic nervous system is the one that, fear, fight, exercise, rage. So it makes sense that it has a relationship with the adrenal gland that produces adrenaline. So let's talk a little bit about the anatomy of the sympathetic division and the sympathetic pathway. So it's also called the thoracolumbar division because its preganglionic neurons are in the gray matter of the spinal cord from T1 to L2. The preganglionic axons leave the cord in the ventral root, enter the spinal nerve, and then pass through the ramus communicans, or small communicating branch, and to enter the sympathetic trunk ganglion. The sympathetic trunk or sympathetic chain lies alongside the vertebral column on each side. After it reaches the ganglion, the axon synapses with a second ganglionic neuron and the sympathetic chain at the same level or a different level, and the postganglionic axon that re-enters the spinal nerve to travel to the skin. Or the preganglionic axon may pass through the ganglion without synapsing and form part of the splenic nerves. The splenic nerves travel to the viscera or to synapse with a ganglionic neuron found in the collateral ganglion anterior to the vertebral column. The major collateral ganglia, the celiac and superior inferior mesenteric ganglia, supply the abdomen and abdominal and pelvic organs, and the postganglionic axon then leaves the collateral ganglion and travels to serve the nearby visceral organ. So review this, not going to be on your quiz, not going to be on your final. But let's talk a little bit more about the autonomic functioning in detail. Again, the autonomic nervous system is broken down into the sympathetic and the parasympathetic. So parasympathetic is your fight or flight. So when we're excited or find ourselves in emergency or threatening situations, or we perceive a threat. So a threat doesn't even have to be real. We just perceive it so. Pounding heart, rapid deep breathing, cold sweaty skin, dilated eye pupils. Under such conditions, the sympathetic nervous system increases heart rate, blood pressure, and blood glucose levels, dilating the bronchioles of the lungs, and brings that about many other effects that help the individual cope with the stressor. So other examples are dilation of blood vessels and skeletal muscles so that we can run faster and fight better, and withdrawal of blood from the digestive organs so that the bulk of the blood can be used to serve the heart, brain, and skeletal muscles. Because when you're in a situation of perceived threat, bear in the woods, your body's not too concerned with digesting your lunch. The sympathetic nervous system is working at full speed not only when you're emotionally upset, but when you're physically stressed. For example, if you just had surgery or you run a marathon, your adrenal glands, which are activated by the sympathetic nervous system, will be pumping out epinephrine and norepinephrine. And the effects of the activation of the system continue for several minutes until its hormones are destroyed by the liver. Thus, although the sympathetic nerve impulses themselves may act only briefly, the hormonal effects they provoke may linger, and the widespread and prolonged effects of sympathetic activation help explain why we need time to calm down after an extremely stressful situation. The sympathetic division generates a head of steam that enables the body to cope rapidly and vigorously with situations that threaten homeostasis. Its function is to provide the best conditions for responding to some threat, whether the best response is to run, to see better, or to think more clearly. So I like to call this fight, flight, or freeze. Some people will freeze in the situation, which is not ideal. And then your parasympathetic nervous system, is your rest and digest system, chiefly concerned with just promoting normal digestion, elimination of feces and urine, conserving body energy, decreasing demands on the cardiovascular system, best illustrated by a person who relaxes after a meal and reads the newspaper. Blood pressure and heart rate and respiratory rates are regulated at low normal levels. Digestive tract is actively digesting food and skin is warm. Pupils are constricted to protect the retinas from excessive damaging light, and the lenses of the eyes are set for close vision. It's also known as the housekeeping system of the body. It is important to know that there can be overstimulation of the parasympathetic nervous system, which results in coma. So an easier way to remember the roles of the two ANS division is to think of the parasympathetic division as the D, digestion, defecation, diuresis, so urination, and the sympathetic division as the E, exercise, excitement, emergency, embarrassment. So developmental aspects of the nervous system. Your nervous system forms in that first month of embryonic development, so fairly early. That means like maternal infections in early pregnancy can have pretty harmful neuro effects on a fetus. One of the last areas to develop is the hypothalamus, and that's why a child that's born premature has a hard time regulating their own temperature. The nervous system grows and matures during childhood largely through myelination, and neuromuscular coordination progresses in a superior to inferior direction and in a proximal to distal direction, with myelination occurring in the same sequence. So the brain reaches its maximum weight as a young adult. Over the next however many decades, neurons are damaged and die, and the brain weight and volume steadily decline. However, a nearly unlimited number of neural pathways is always available and ready to be developed, allowing us to continue to learn throughout life. So we just have to give ourselves and our patients stimulating activities to keep that brain function up. As we grow older, the sympathetic nervous system gradually becomes less and less efficient, particularly in its ability to constrict blood vessels. So when older people stand up quickly, they often become lightheaded or faint. The reason is that the sympathetic nervous system is not able to react quickly enough to counteract the pull of gravity by activating the vasoconstrictor fibers, and the blood pools in the feet. This condition is called orthostatic hypotension. It's a type of low blood pressure resulting from changes in body position. The usual cause of nervous system deterioration overall is circulatory system problems. For example, arteriosclerosis, which is decreased elasticity of the arteries, and high blood pressure reduce the supply of oxygen to brain neurons. A gradual decline of oxygen due to the aging process can lead to senility, or somebody that is senile, characterized by forgetfulness, irritability, difficulty in concentrating and thinking clearly, and confusion. A sudden loss of blood and oxygen delivery to the brain results in a CVA, a stroke, as we talked about earlier. However, many people continue to enjoy intellectual lives and mentally demanding tasks their entire lives. In fact, fewer than 5% of people over the 65 demonstrate true senility. Sadly, many cases of quote-unquote reversible senility are caused by certain drugs, low blood pressure, constipation, poor nutrition, depression, dehydration, and hormone imbalances, and it goes undiagnosed. The best way to maintain your mental abilities in older age is just to seek regular medical checkups throughout life. Although eventual shrinking of the brain is normal, some individuals, boxers, alcoholics, they can hasten the process. So the likelihood of brain damage is higher, atrophy increases with your boxer with every blow. The symptoms of slurred speech, tremors, abnormal gait, and dementia are seen in many retired boxers, and there's pretty much resounding evidence that alcohol has a profound effect on the mind as well as the body. So CT scans of chronic alcoholics reveal reduced brain size at a fairly early age. Like boxers, chronic alcoholics tend to exhibit signs of mental deterioration unrelated to the aging process. All right, you guys, I know that was a lot. Any questions, comments, or concerns, please feel free to let me know.